And thank you so much, Dr. Walia, for agreeing to participate, especially during such an unprecedented time. And I will like to introduce Dr. Walia. So Dr. Walia has received his PhD in plant biology from the University of California, Riverside. His research interest is in understanding the physiological and molecular basis of abiotic stress tolerance in crops. His program uses genomics and phenomics approaches for discovering novel genes and genetic variants for improving crop performance in suboptimal growing conditions. Of particular interest are crop responses to variation in water quantity and quality and high temperature. So if we could give a virtual applause to introduce Dr. Walia, the mic is over to you.
Yeah, thank you, Karuna, for the invitation, and good morning, everyone. It's my pleasure to be able to share some of the work that we've been doing. As Karuna mentioned, that my interest is in understanding how crops respond to their environmental changes, and of great interest are water and temperature. And given that we have the climate change context, there is really some urgency in understanding how crops can cope better with these changes in environment. So today I'm going to focus on our work on temperature. And when we think of temperature, we usually think of blazing sun and really hot and sweaty conditions. And I know in Canada it's not that often. But so what I've talked today, share with some results with you, is about the nighttime temperatures. So, and the reason is, if you look at these two maps, you have average daytime temperature shown here on the map on the left, which is what it shows is the change in average annual temperature or maximum temperature for the last 110 or so years. And so more red means that the temperatures have increased. And if you see blue, it means the temperature, average temperature, maximum temperature has decreased. When you separate it from the night temperature on the right side, you see a lot more red. What that indicates is that globally, the nights have become warmer, more warmer than the day temperature. So, and the nighttime changes or increase in nighttime temperature, which we, in our group and in several other groups, we call it high night temperature. So HNT for high night temperature stress. So if you look at, how in North America, you see a large swath of, red that's indicating quite high temperatures around the Hudson Bay, so it's kind of spreading out a lot more as compared to the high day temperature changes. Of particular interest is the how crops respond to these temperatures. So if we look within the U.S., what's been striking is that this is on the left side again is the average maximum temperature, so that would be typically daytime, and on the right is the average minimum temperature. So any anomalies from a normal temperature are shown as these bar graphs. So the red here indicates higher temperatures, and blue indicates anomalous years that are cooler than the regular average. So what's been striking here is that the most of your red high bars are, or in some of the warmest years, have been since 2000. So clearly there's been an uptick of temperature both for average, maximum and minimum, but more prevalent in the minimum side on the right graph. So based on the NOAA's calculations, in the U.S. the nights have warmed up at twice the rate. So there's about a 1.4 degree Fahrenheit increase in nighttime temperature compared to about 0.7 degrees Fahrenheit in daytime temperature. So this becomes relevant not only for North America and US, as I showed on the global map, that there's several studies that have now clearly indicated that the many regions that grow rice, which is what I have studied, along with wheat, have had much higher rate of increase in nighttime temperature. So, and why this is important is kind of shown here in this graph. If you combine the output from rice and wheat, in combination provides more than 50% of the calories that humans consume per capita globally. So the fact that, for every 1 degree increase in the average nighttime temperature during the growing season for rice, we see a decline in about 10%. And this is field-based data. So it's almost alarming that, you're seeing these great increases in nighttime temperature. For wheat, which I'm not going to talk about, but I would love to at some point, we see about 4 to 6% decrease in yield for every 1 degree increase in nighttime temperature. So for rice, it's not just only about the yield, but it's also about the milling quality, because rice has these higher nighttime temperature. Rice has this problem where it makes more inferior grains, and inferior grains, when they undergo milling, where rice gets polished for selling to stores and consumers, it starts to the grain becomes very brittle and it starts to break. So that's one of the concerns. The second concern is that the hybrid rice or the super rice, which is addressing a lot of the food needs, increased food demand for rice, is supposed to be also very susceptible to heat stress in general and higher nighttime temperature. So increasingly, the rice grown in the U.S. is hybrid rice, and a lot of rice grown in China is also hybrid rice. So it really poses a bigger threat to food security as well as food quality. So before I kind of dive into the details of what we're doing, I do want to kind of give you a physiological perspective of what we're talking about here. So this is a rice panicle. So the inflorescence of rice, which bears the flower and then the seed, is called the panicle. If you look at the grains at the very top and the grains at the very bottom, that tend to fertilize later and hence have poor quality and smaller size, are called the inferior grains. And most of the grains in the middle tend to be well filled and of higher quality. So what high night temperature does is to rice is no different from most of the other organisms, including humans, is that increased temperatures increase the rate of respiration, which results in loss of carbon that the plants have fixed in form of carbon dioxide. But at nighttime, that loss is not as well compensated as a higher daytime temperature would. So that kind of result, that's one of the hypotheses. The second is actually been observed by several groups, is that the rate of starch accumulation in the grains is affected by higher nighttime temperature. And of course, the superior grain and the inferior grain have a differential response to high temperatures. So those have been well documented. But what's not been done or recorded so well so far is the genetics behind it. So what we wanted to do was take a very holistic approach in addressing this problem of higher nighttime temperature stress. And for that, we had a joint project with we have a joint project with a group of researchers at Nebraska, in Kansas, and Arkansas, and Rice Tech is also a partner, which is the largest hybrid rice seed breeding company in the US. And what we did was we used this layered approach. So we wanted to explore the gene space because you want to look at the genes and make discoveries that could be useful for breeders. And then we also wanted to look at the transcript space where some genes are expressed and that the level of their expression could alter the response that individual genotype would have for a high night temperature. And given that we have the seed quality issue, we also wanted to dive into the metabolomics. And then finally, all of this is just as good as as your phenotyping. So we wanted to do a phenotyping approach that goes from a single plant in a pot in a greenhouse to plot level. So then be able to combine all of these information to come up with not only genes that we can suggest as possible sources for higher night temperature tolerance, but also develop methods and of going across scales and seeing what we find. So this is Valerio asked me about if I do field studies. So the answer is no, but I do work quite closely with collaborators. So this is the field site on Rice Tech's field research station that my colleagues in Arkansas have kind of developed, where we have the infrastructure to, take these really heavy, big tents. We pull them in once the rice is about to flower, and then we open them up, unroll, roll the tall canopy off during the daytime. But then at nighttime, we drop them down, and then we heat these tents by 3 or 4 degrees. So what I'm going to talk. So we've got this 2 seasons worth of effort in the field, and we're looking at the data. It's a lot of data, but what I want to talk today mainly about is what we did in the greenhouse. So before I dive in, I just want to give you a brief overview of rice grain development. I know most of you are probably thinking of introductory biology right now, but basically, this is the rice grain. You have the embryo, which is about 8%, and of dry weight, and it's the maternal and paternal tissue, and then you have the maternal husk tissue, which is just the mother's genome, and of course, endosperm is quite intriguing and the most important organ. In some ways for plants. So it's made up of 2 mothers' genome and 1 father. So that kind of, once you have this fertilization in embryo to make embryo and endosperm, that's the double fertilization. So once the fertilization happens, the seed goes through this rapid increase in size. That's kind of shown here as black in this cartoon, as the black is what the color represents the seed, and it grows rapidly. And then once it reaches that size, that's at least the length and length in terms of length, the size that it's going to have, and then it starts to grain fill, and then there's a dry down period. So this window, my lab's been very interested in understanding this particular window and how temperature affects that, because that's a stage where we think that rice and wheat are both, and I'm sure other cereals too, are very, very sensitive to temperature. So with this overview, I'm going to kind of share the experimental design for this, for the work that I'm going to talk about today. So what we did was we took about 300 rice accessions from the Rice Diversity Panel 1. So these are accessions from, I think, more than 80 countries. So they're very diverse, land races, cultivars, and so on. And we grow them. And once individual under control optimal conditions, so that's 30 degrees during the daytime and 23 at nighttime. And then once these plants start to flower, we mark them. So when we have sufficient flowers on any individual panicle, we mark them. And then 1 day after fertilization, so we want that embryo and endosperm double fertilization to get established. And then we move a subset of the plants into another greenhouse where the daytime temperature is still 30 degrees centigrade, but the nighttime temperatures have been increased to 28 degrees. And our greenhouses maintain these temperatures very well. And once that sort of sets off the series of almost back-breaking experiments where transitions where you have to kind of monitor 300 lines that don't flower at the same time, have different heights and so on. But besides the logistics, what we did was we took these plants out of the greenhouses, for a few minutes, and we imaged them every week. So we image them for week 1, week 2, and week 3, and then we harvest the plants at maturity, which is about a month, about 30 to 40 days for most accessions. So in this process, we ended up imaging about 1,800 panicles, individual panicles that we had marked 3 times. And of course, at maturity, we also looked at panicle weight, grain length, grain width, and grain weight. So the first part of my talk is going to be on the imaging side, and the second one is going to be on the maturity phenotypes. So how did we do the Panicle imaging? So we worked with my colleague Hongfeng Yu and his two graduate students, Fei Yu Zhu and Tian, who developed an imaging box for the Panicle imaging. So here's the animation of what it looks like, and then I'll show you the real one. But the context here is to see the superior and the inferior spikelets and be able to do that. So what we do is, since the plants have different height, we built this pedestal that we can raise up and down, and then there's two cameras at slightly different angles go in and take 60 images each within a minute, and then we remove the plant and we put the next plant. So we did that about 1,800 times during this experiment. And this is well, this is what the real setup looks like. You only see the Panicle here because the rest of the plants kind of, there's a good 4 or 5 feet underneath where the rest of the plants there, and it has to be done very gently so that we don't break, because we have to image the same Panicle for the subsequent weeks and then harvest for maturity. So this is what it looks like. The two cameras are programmed that we with in-house script that they would take these images. They are at two different angles to prevent occlusion, and I'll come to in the next slide I'll show you why we did that. So we take 120 images, and what we did next was, typically you would take images, and you normally don't need 120 images, but we would do a 2D image analysis, which is shown on the right side. Basically take a flat image, RGB image, and we segment it, and we count the total. Segmenting is like separating the plant part from the background, and then we count the total number of pixels. So that was nice and good. It's fairly straightforward these days in phenomics, unless you have very complex architecture. But what we did next was that we also tried to create 3D reconstructions. So by having all these images from all sides and at different angles, we are able to create a volume, a 3D space, in the basically a volume in a surface area. So what that is called is basically a point cloud, which we segment out, and then we start to extract features such as different colors, R pixels versus G pixels versus B pixels. So RGB would be the 3 colored pixels from the camera, and we also can calculate the surface area and the volume. So this all we published this work last year, I think, and all the code's available in GitHub. I think there's a possibility of actually changing a little bit and being able to use it. If anybody's interested, we'd be happy to help them. Or if you have any questions to address that. But before I start selling that, I should probably show you the data. So what we did next was we had this 3D voxel count, which basically would give you a volume as opposed to a 2D surface area of developing panicles, and W1, W2, and W3 are weeks 1, 2, and 3. And we then looked at the correlations at maturity of the same panicles that we had imaged for panicle weight, seed number, and panicle area. What we found was quite striking. So if you look at the correlation at week 1, it's 0.48, but as the panicle development matures, or progresses, you get up to 0.74 in terms of correlation. Whereas if you look at just the 120 images and the information that you extracted from those 2D surface area images, you don't see nearly as good correlations. So what that kind of implies is that the 3D reconstruction provides us with a much more accurate proxy for what the mature phenotypes would look like, as opposed to just having a stack of images that we just kind of segment through to get an average value for pixel. Similarly, for Panicle area, which we calculated by scanning the mature harvested panicles in a flatbed scanner, there's is also has high correlation with your by week 3. So that clearly showed us that we can reliably do this on a large scale, very backbreaking in some ways, because and sometimes heartbreaking when you actually break the panicle while doing this. But it's doable. So one instance of what does that mean, when we compared a small subset of lines from control, shown here in blue, and HNT stress in gray, we start to see a difference in voxel count, which is the volume in the 3D space by week 1 already, and then it sort of catches up to by week 2 or week 3. So we can pick up differences very early, which you may not be able to pick up in terms of phenotypes or traits if you were harvesting at maturity alone. So we also were able to look at certain factors, such as looking at just the ratio of R pixels to G pixels, because as the panicle matures, it's going to turn from green to brown, which means that most of the pixels are going to transition from having the G color count to R color count. So when we look at the ratio of R to G, what we found was that we were able to also report that the maturity under HNT condition is accelerated as the HNT stress progresses. So that's kind of consistent for several gene accessions. I think 30 accessions here, compared to what people have already reported, that you see a difference in panicle maturity / senescence with this with high-end HNT stress. So what we did next was we applied this. We extracted all these traits that were not, and then we picked up the ones that were normally distributed for the entire panel of lines, and then we started to we did GWAS. When we do GWAS, let's say for week 1, we found several candidates or peaks. So what this is showing here is a Manhattan plot, which has the 12 rice chromosomes. And these dots are individual SNPs, and the higher they are or more stacked they are, and plus higher, means there's a higher chance of a significant association between polymorphism in DNA and the trait that we are measuring. So in this case, we looked at the average green pixel value at week 1, which should kind of give us a sense of the biomass. And what we found in one instance was, and there's several big peaks, but in this case, we found that SPED1 was one of the top SNPs is located in SPED1. That's the gene that's been reported previously in rice to regulate the pedicel length. So if you have a mutation in this gene, this is 9311, which is a wild type. If you have a mutation in this gene, you get these pedicels that are like much shorter. So the panicle is actually more compact, which is shown here on the image on the left side. That this is a regular wild type panicle, and this is the one that has the shortened pedicel, so hence a more compact. So our imaging is actually able to pick, and this is just one instance, but our imaging is able to pick these instances. Basically genes that are known to impact panicle architecture, but we can find that there's a genetic variation for this gene in the rice germplasm. So what we did next kind of brings back to the idea that I talked about, the superior and the inferior. So what we did next was we actually asked if we can, we have this 3D point cloud carved out in space. Could we actually slice it? So we arbitrarily sliced it into 10 different pieces. The idea being that the superior grains would be kind of in the center, and the inferior grains would maybe in the top, and definitely in the bottom side, because they're developing when the carbon resources are being plunged under a little bit more strain because of HNT stress. So we did that, and this animation kind of gives you an idea. So, but what it looks like, I also wanted to show you this, is it's green largely for week 1, and you see for week 2, as the grains start to fill, each of those branches become heavier, so they start to kind of pull away from the main axis. And then you also have the week 3, when most of the greens turned into brown, so you'll have more red pixels as opposed to green pixels in week 3. So this is kind of I think shown in this animation, that so these are point clouds, even though I'm showing you in 2D again. But you can actually rotate and get a very good perspective of what the thing looks like. So we sliced it, and we looked at, many traits were not normally distributed, so we didn't want to worry about it at the present analysis. So we worked with normally distributed phenotypic traits, and then we did some genetic correlations. So each of the slice from the bottom to up is hist 1, hist 2, hist 3 for histogram 1, 2, 3. That's how we started looking at it. So I don't know if that's the best terminology, but what came across was quite striking, that this is the color heat map for correlation. Deep red and deep blue would mean high positive and negative correlation respectively, and whitish would mean that it's not a very strong correlation. What we found was that for instance, when we look at the red pixel count for control, we can sort of see a clear demarcation from hist 1 all the way to hist 5. So hist 1 is more highly correlated with 2, 3, 4, and 5. And then you see a second section where you have high negative correlation between hist 1 and the middle part of the panicle, and then that correlation starts to erode again in the very top of the panicle. And we see that if you impose HNT stress, that difference is actually not that big. So it seems to, theoretically, and I'm kind of going on very thin ice, is that the superior grain might be getting more affected by HNT stress and starting to look more inferior. Hence, you don't see as big as deep a color-based demarcation. Color being not the pixel color, but the correlation color between the ends of the panicle and the middle. So what we did next was we took each of these segments and the traits that we derived from them, and then we performed GWAS. So I wanted to just give you a sense of the kind of things that you see. There's many significant ones that we are trying to follow, if they're not just transposons. But you see these correlations for hist 4 and 5. For some peaks would persist, let's say, on hist 4 and 5 and 6, but others would be, let's say, this green one here is on hist 5, but you don't see as much significance at that loci for 6, but again, it emerges at 7. So and this is just week 2. So if you start thinking about it, the response that really matters for HNT stress in rice is the panicle and how it behaves. You're getting these differential response not only over time, so you might have genes that are important for HNT stress tolerance or sensitive or leading to sensitivity, allelic variation in the first week, but are not evident at 3rd week and may actually have no detection by the time you do the mature phenotype. So it starts to show that there's this space when you're starting to look at environmental stress, where you have this spatial and temporal component, and there's a genetic basis that kind of lies behind it. We're still digging into it, but I wanted to give you something, give some sense. So one of the peaks that my postdocs, Puneet Paul's, kind of detected, it's not the most significant peak, is under HNT stress for a MADS transcription factor called MAD79. So Puneet's work published last year, or actually this year, he showed that MAD79 is involved in seed size and quality. So he was really interested in this. So now what he's doing, since he saw that one of the candidate SNPs falls within this gene, So he's actually looking at the knockouts, CRISPR knockouts for this gene. And there's at least the first replicator we have, it's fairly significant number of plants. It shows that this knockout does impact both the seed size and seed weight under HNT stress, and also HDNT stress, which is basically both higher day and night temperature. So, and we're imaging as I speak every other day for the whole month for these mutants. So hopefully we'll see some evidence that kind of shows that not only the mature phenotype, but actually the developing phenotype that we're picking up here for this is important. So now I want to transition to, this is all imaging. It's good. It was more discovery, some of it which we didn't expect to work out, but it really does open up some new insights into how HNT and this temporal spatial component of stress response in plants. So what we wanted to, the main plan was to kind of look at the mature phenotypes, and we looked at different phenotypes here, like panicle weights and seed lengths, seed weight per seed, and so on. And there's some correlations that most of them are between control and HNT, and most of them are, as expected. Some things change. But what we did next was we performed a traditional GWAS analysis, again, going through each SNP and asking if there's a correlation, whether the SNP has one polymorphism or the other, or the other variant, and trying to correlate it with phenotypes. So when we looked at grain length surprised phenotype in rice. What we found was that GS3, which is a very well-known, maybe at least 10 different labs have kind of narrowed down on this gene, is a major SNP for grain length. And it's kind of evident when this is all the 12 rice chromosomes. I made it more colorful this time. And of course, under HNT stress, you also see the same gene. There's other genes, SNPs, that are not shown here, that don't emerge from the control conditions, but do emerge from the HNT. So similarly, we also looked at grain width. And what was interesting here is that several of the genes again, or the SNPs again, were controlling grain width under control and HNT stress, but there were some unique ones that we didn't find in the control conditions. So that was kind of striking, because if HNT is a difficult stress to impose, or heat stress in general is a difficult stress to impose in field conditions. So if you're breeding for that heat tolerance, and you're pretty much counting on a location or a hot year, a hotter location or hot year. So you might be missing out on some of these genes or alleles that could be important for HNT tolerance. So we've focused on we're actually working on all three of these, HNT 5.1, 8.1, and 8.2, but I have the most results for HNT 8.1, so I'm going to speak about that. So it's a single SNP. It's only seen under HNT stress. We don't have any evidence of any SNP that emerges from the control, and we've tried different ways just to make sure we don't miss. The HNT 8.1 lies within a gene called fertilization independent endosperm 1, or PHI1. So it lies in the intron of this gene, and so it was of great interest to us because PHI1 is a member of a polycomb repressive complex group. So polycomb repressive complex 2, or PRC2 in short, is known to regulate the early development of endosperm. So this is the same developmental stage which I was showing you in that early, introductory biology double fertilization cartoon. So this gene, what we found was there's several members of the PRC complex that are shown here. And what we had earlier work in my lab long before this project started had shown that this was the only gene at transcript level that was responsive to heat stress. So it's induced earlier by heat stress. And what we found, so that's why it was interesting for us, plus the fact that it was an HNT specific gene. So what we find is that if you have the major allele, in blue is your control conditions, in orange is your HNT, the major allele, which is the big M, have more wider grains. And if you have the minor alleles, you have less wide grains. So we looked at the HN51 response to heat stress, and what was very striking, and the reason why we wanted to work on this a little bit more, is that it's not that so this is heat stress and this is control in green. So it's not expressed during the early stages, the first 24 hours, but right around 36 to 48 hours, the expression of 5.1 goes much higher under heat stress. The other striking thing that I should share is that when we look at 48 hours after fertilization and we increase the temperature by 2 degrees, so you go from 28 to 30 to 32 to 34. So the temperature, so the expression of 5.1 actually responds to every 2-degree increment in temperature. So those were like the background information that we already had from like work done 6-7 years ago in my lab, and so this is what we started to look at under HNT stress. So these are some 4 of the lines that the big M1, M2, M3 are the major alleles, and small M1, M2, M3, M4 are the minor alleles. And you can see how the differences in temperature, you see a big difference here in M2, the small M2. So we asked if we looked at 4 days and 7 days after under control and HNT condition, and we asked how the gene expression changes in these major and minor alleles. So what we found was that the transcript abundance of 5.1 increases by in many of the cases, or at least one of the cases in at 4 days, but definitely in all the 7-day samples in the major allele, the expression is higher. Whereas in all the minor alleles, the 5.1 expression is suppressed by HNT stress at these 2 time points. So that sort of helped us set the hypothesis that maybe transcript abundance of this gene is important. So we created CRISPR knockout mutants for these for 5.1, and we looked at the mature grain length and grain weight and single grain. Again, the blue here is controls, wild type is WT, and I show the results from 2 of our CRISPRs. We have several more, but we focused on these so that we can kind of do large numbers and with smaller number of lines. So what we find is that the grain length, decreases a little bit, but it's not as sensitive, which we've also seen in many other accessions. Excuse me, I think my computer's going a little bit. And then the width, however, increases. So if you have a control CRISP, if you knock this gene out, basically under control conditions, the width it's much wider. However, if you look at the heat HNT stress response, in orange, you see a significant drop in the grain weight, and even more so in single grain weight. So that was kind of, it was good to see that we can, the 5.1's doing this under HNT stress response, but it was kind of striking that you actually get maybe not supremely bigger seeds, but you actually see bigger, more wider seeds, but they weigh less. And so we wanted to see what's going on. So we actually looked at these grains a little bit more carefully. So this is on the left 2 panels for control and HNT stress are just basically light box scans. So for wild type controls, kind of clear, but HNT stress, and this is what I was talking about, like the grain becoming inferior, is because you start seeing the browning in here. And then if you look at the CRISPRs, you actually see a little bit of browning already, even under control conditions, but you see a lot more of the opaqueness in the HNT stress. So then we followed up with the same seeds, same set of seeds for SEM microscopy. So what you see is under control, you get this starch structure that's got this very well-defined, crystal hexagonal structure. But this is what HNT stress is doing to the rice starch deposition. So it creates these globular structures with lots of air packets that really doesn't, basically the light doesn't go through or gets transmits as well. So that's why you start seeing those opaqueness in the light box. And once you mutate this gene, 5.1, you basically see this puffy mess that looks very similar to HNT stress response, and it just gets worse when you impose HNT stress. So even under control conditions, the 5.1 mutants have major issues with starch deposition and starch packaging that just get aggravated under HNT stress. So we then asked if this was also true for the major and minor alleles. So here are the M1, M2, M3, M4. These are the more tolerant, have the tolerant allele for 5.1. So their grain is heavier and wider under HNT stress compared to the minor alleles. So in this case, again, you see some effect, but not as prominent. But if you look at the minor alleles, with the exception of the small M1, M2, M3, and M4 have really bad starch packaging issues going on. So this kind of sort of indicated that 5.1 could be an important factor in maintaining rice grain starch packaging, at least, and also rice seed size under HNT stress, and seeds that are deficient in that. So the minor alleles actually have a down regulation of this gene under HNT stress. So as they become deficient, they become more prone to turning into inferior quality grains. We also looked at the sensitivity of these mutants under high day and night temperature, because high day, warmer days are moderately correlated with warmer nights. So we don't see much of a difference in grain length. But if you look at the grain width, so these are under both hot day and night temperature, we see a major drop in the seeds that are deficient in 5.1. And also similarly for single grain weight, the grains are just, so what's happening is, at least my thought is that what's happening is that even though the seeds look a little bit bigger, the wider specifically, but they're puffier. So it's like they're being popcorned. The starch is being popcorned within it under heat stress when you have deficiency of 5.1. So kind of to summarize the results, so we're basically characterizing 5.1 in more detail and what's the underlying transcription factors, given that 5.1's epigenetic regulator for endosperm. So my main takeaways from today's talk would be that the natural variation for grain yield parameters under high temperature stress is partly regulated by loci that are not very evident when you're screening for your materials under optimal conditions. We would be a lot more sure about it once we have analyzed all the field data to support or not support what we're finding from the controlled environment conditions. The second takeaway is that the 3D imaging, while very laborious, may provide mechanistic and genetic insights that are not going to be forthcoming if we were just working with mature trait phenotypes. Of course, there is a good solid argument to be made that if you don't see it in the end, and there is a counter argument for that. If you don't see it as a terminal phenotype, which is seed size or grain weight or something, then does it matter? And then of course the allelic variants within 5.1 impact both the grain width under HNT stress and normal starch deposition. We also, one of my students working on more detailed fluorescence microscopy, and she's seeing evidence that the protein and the lipids are altered. So the starch quantity is not altered, but the starch packaging is, but that somehow impacts the protein and lipid levels within the mutations, within the mutant lines. So we're still kind of figuring out, working our way through the data, but there's definitely some significant changes there. So with this, I want to acknowledge not all, but an enormous effort that's gone in from people in my group, Puneet, Jaspreet, Balpreet, and Larissa and Martha Rowe, have worked and continue to kind of process a large number of seeds and lots of imaging. Professor Paul Stasvik, my colleague here, helped develop the cabinet for imaging. And then Hongfeng Yu in computer science and his 2 students did all the image analysis, 3D reconstruction. And then students and postdocs in Dr. Gupta Marota's lab in Virginia Tech did all the GWAS, especially the spatial temporal GWAS, which was kind of, we couldn't find any existing methods for that. And so they're still under development. And then Argelia Lorenz's group in Arkansas State and Arlene Adviento at USDA are kind of involved in, they're the people in charge of the tent, HNT tents in Arkansas. And thankful for funding for this work from NSF. And I'd be happy to take any questions and any comments or thoughts or ideas are all very welcome. Thank you.
I think we can all give a virtual applause. Thank you so much, Dr. Walia. Appreciate it. I think it's a very interesting approach of amalgamating the imaging and the phenomics and the genomics, and that can provide us really good insight. So I think we're ready for the question answers round, and I would appreciate if somebody can initiate the first question. I think Dr. Singh is here. So,
can I ask a question, Karuna?
Yes, sir.
Yes, please.
Harkamal, this is a great seminar. I really enjoyed it. It's wonderful work. So I have a very quick question about about your SPAD genes. SPAD1 you mentioned, which you have identified through genome selection, right? Sorry, GWAS, right? Yeah. So it relates to the architecture, spike architecture in rice. I'm just wondering if how similar it is with the VRS1 gene, which was identified in barley for the 6-row versus 2-row. Is there any information, or it is totally different from that?
I don't know. I didn't check, but I will certainly check as soon as we get done here. That's a good point. Like, I didn't actually, SPED1, the mutation itself was characterized previously by a group from China in 2014. What we showed was that as a basis of validation for our image analysis based GWAS is that we can pick SNPs that actually, I think this particular SNP is next to or inside SPED1, and that it also explains natural variation for the Panicle architecture of branching in rice germplasm. So my short answer is I don't know. I did not look at the VRS1 and SPED1.
Yeah, chances are pretty high because the VRS1 is doing the similar type of things, and I think that will be a great opportunity. Maybe we can think about something together, looking at that because this is a really an important domestication trait, right? So if somehow there are some relationships there.
Yeah, and I think I should have mentioned is that the same imaging system works quite well for wheat, especially if the wheat's not on. If the wheat's not on, it actually works better for wheat than it does for rice, just because it's more compact, the inflorescence, so the spike is more compact. So yeah, so definitely if there's some interest in pursuing this in barley or wheat, we'd be happy to. Wheat would be very easy for us because we're doing this for our wheat diversity panel, or we've done this for a wheat diversity panel as well. And barley also, I think, would not be that different, especially if it's on less barley.
Okay, thank you.
Thank you, Dr. Singh. Is there any other question? You can also write that down in the chats, and I can read that out for you.
Yeah, I have a question. When you did when you found that starch deposition was changing due to HNT stress, okay, that the conformation of the starch granules was significantly changing. Have you had any thoughts as to what is going on with protein as well?
So yeah, the results that I showed you were just for the packaging of starch. But what we are currently, one of my PhD students, Balpreet Dutt, she's looking at she's using fluorescent labels for starch and proteins and lipids separately. And what she's seeing is that the starch levels are not changing, the packaging is changing, but the protein level for HDNT stress and to some degree HNT stress is going up by at least for sure at 7 days after fertilization, and similar observation is there for lipids. So the protein is actually increasing. Starch is not decreasing significantly. It's just the depositions are different, but proteins are definitely the protein content of the HNT stressed grains is increasing.
Right, thank you. And the second question I had was, I don't really know exactly how to ask this question, but so I understand that heat stress, because heat and drought, they're correlated, right? And there's not exactly the same type of stress, but like the response in the plant is somewhat correlated. And so how do you reconcile the fact that you have that heat stress during anthesis? Like, what's the relationship with yield here, right? Because I'm assuming that you're trying to get at yield reductions under heat stress.
Mm-hmm.
So what is the plan of reconciling the anthesis period under heat stress and then starch deposition from the fact that heat stress, the yield is dropping under heat stress from basically floral abortion a lot greater rate rather than starch deposition, if that makes sense. Starch deposition is under the assumption that that the plant was successful at extruding anthers and then going through the whole floral process.
Yeah, so I think that's a good question. I think the way we address this in the experimental design, not address your question, but an aspect of it, is that we initiated this terminal HNT stress 1 day after fertilization. What that does is, for most genotypes, it would not result in any striking difference in fertility, because we are waiting and we're marking. So that's what's time consuming and laborious, is that we focus on the primary panicle. We let a whole bunch of them on the day that a whole bunch of them fertilize. They take about 3 days to go through the whole panicle in terms of fertilization. And then we start marking, and so we followed individual genes. Not in this case. In this case, we looked at the whole, but we typically follow individual genes, individual seeds that we have marked, and we don't see a drop in fertility for most genotypes. However, what you're pointing to is that when we start to look at the entire yield from multiple panicles, because rice has tillers, so it has multiple seed-bearing panicles. Some of them will experience the HNT start to experience the HNT stress before they have fertilized, and others will be well along the way. So that's something that we are going to see from our field studies, where where we will have that, basically a differential in when the stress is imposed. So what we do know is that some indica lines tend to, not all, but some. So there's but at least 3 or 4 indica lines offer a set of 12 lines that we worked very in greater detail, trying to get to some of the questions that you're asking. Some of those indica lines would abort the seed and make bigger seeds. So some seeds would get aborted, so the carbon will actually flow into the remaining seed and they'll make bigger seeds. And some of the temperate japonica lines, so japonicas have both tropical and temperate, would tend to have very high fertility and also, but make smaller seeds. So we'll make more seeds and smaller versus the, in cases of Aus and Indica, which are climates where you would actually see a lot more of heat stress in general. And I don't want to generalize by saying Indica, but that's what we've seen from a smaller set of lines, that where you have more aborted seed and then you have pretty much a resilient seed size. And that's, of course, rice can, one thing that you also see is that in many accessions of rice, when you have HNT stress, they start to make more panicles, seed-bearing panicles. And so there's all kinds of, dynamics of source and sink that are happening. But that's another thing that would be pretty hard to characterize in some ways. But I'm hoping that some of that is what we would address in the future. But I don't think we can go as precise as the data I'm showing you within one panicle, really high resolution, marked carefully, because then you're looking at 10 or 12 panicles within a single plant in many cases.
It's a yield component compensation problem. Yep.
Yeah.
Thank you. It's a very good answer.
Do we have any other questions? Okay, I guess then that will be the end of this session. Thank you so much, Dr. Walia. I really appreciate of you agreeing for participating in our Research Horizon series this semester.
Yeah, thank you for the invitation. It was my pleasure.
You're very welcome. Okay.
All right. Thank you, everyone.
Thanks.
Bye.
And this concludes our Research Horizons seminar series for the semester. Hope everybody has a good break, and Professor Jabaji will be coordinating next semester's Research Horizons, but so we hope to see you all at our seminars. Don't forget to fill out evaluation forms, Mercury evaluation forms, and provide us feedback on how we ran the seminars this semester. It helps us, it helps us improve, and also our for the student series, please don't forget to fill those out or submit them if you haven't done so. Okay, have a good break, everybody.
Yeah, thanks.